have you ever wondered why are we all so different from one another? Well, the answer to that question lies in our DNA. So, what is DNA, you might ask? Well, DNA stands for deoxyribonucleic acid, which is a hereditary molecule that contains some codes or biological information. This information or code is different in all of us, which is why each of us is unique. Who discovered DNA? Well, DNA was first identified in the late 1860s by Swiss chemist Friedrich Miescher. And many years later, in 1951, Rosalind Franklin discovered that the DNA has the helical form, and later in 1953, James Watson and Francis Crick discovered the structure of DNA, for which they were awarded a Nobel Prize in 1962. So, where is DNA found then? Well, DNA is found in nearly all the cells, and cells are broadly classified into two types: prokaryotic cells and eukaryotic cells. While eukaryotes, like we humans, have multiple cells with various compartments. So, DNA in eukaryotic cells is located in one of the compartments called nucleus. On the other hand, prokaryotes, like bacteria, only have a single cell without any nucleus. So the DNA is mostly located in a central part of the cell called nucleoid. So what is DNA made up of then? Well, DNA is made up of building blocks called nucleotides. A nucleotide consists of three components. First, a phosphate group. Second, a carbon sugar. And third, a nitrogen base. In DNA, there are four kinds of bases: adenine, guanine, cytosine, and thymine. Adenine and guanine are also called purines, and each of these has two nitrogen-containing rings, whereas cytosine and thymine are called pyrimidines, and each has a single nitrogen-containing ring. So what is the DNA double helix then? Well, double helix is the term used to describe the structure of DNA. Why? Because DNA looks much like a twisted ladder. Each rung of the ladder is made up of two nitrogen bases paired together by hydrogen bonds. Adenine always pairs with thymine with two hydrogen bonds, and likewise cytosine always pairs with guanine with three hydrogen bonds. The number of such complementary base pairs is often used to determine the length of a double-stranded DNA molecule. The two strands of DNA molecule are antiparallel. One chain is oriented in a 5' to 3' direction, and the other is oriented in 3' to 5' direction. So what does DNA do after all then? Well, DNA contains all the instructions that's needed for an organism's development, survival, and reproduction. To carry out these functions, DNA sequences must be converted into messages that can be used to make proteins, as proteins do most of the work in our body. So the production of protein is vital, and therefore there is an importance of DNA. And that's it for this video. I hope you like it. Please don't forget to like, comment, share, and subscribe.